Good afternoon again. It is a great pleasure to welcome Dr. Richard Roberts to this series of special lectures. Before we begin our special seminar, Assistant Professor Dr. Nippon Tuwanon, President of Chiang Mai University, would like to say a few words to welcome Dr. Richard Roberts. Dr. Nippon, please.
Dr. Richard J. Roberts, Mr. Yanis Hegwai, Director of Events Coordination of the International Peace Foundation, Professor Supot Wuttikan, Dean of the Faculty of Medicine, distinguished guests, ladies and gentlemen. 2004 marks the 40th anniversary of the founding of Chiang Mai University, and I am sure our former president would agree that one of the rare privileges bestowed upon a president is the opportunity to preside over such an auspicious occasion, as having a Nobel Laureate for medicine in our presence. Dr. Roberts, it is indeed an honor for me to welcome you to our university. Dr. Roberts is with us today under the auspices of the Nobel Prize Laureates and Celebrities Lecture Series, a program which will provide our staff and students with a unique opportunity of coming face to face with some of the world's most distinguished scientists and influential figures. A once-in-a-lifetime opportunity that I am sure will be a great source of inspiration. I would like to take this opportunity to express our gratitude to the International Peace Foundation, without whose support today's lecture would not be possible. Also to thank the Dean and his colleagues in the Faculty of Medicine for the time and energy they have contributed to organizing and hosting this event. Dr. Roberts, our most sincere thanks and appreciation to you for honoring our university with your presence. And I trust that the memories you take with you of our university and our city will hasten your return in the not too distant future. Thank you.
Our speaker today, Dr. Richard J. Roberts, is a Nobel Laureate for Physiology or Medicine and a research director at New England Biolabs, a biotechnology company based in Massachusetts. Dr. Roberts was educated in England, attending the University of Sheffield, where he obtained a PhD in organic chemistry in 1968. He did his postdoctoral work in Professor Jack Strominger Laboratory at Harvard University, studying the transfer RNAs that are involved in the biosynthesis of bacterial cell wall. From 1972 to 1992, he worked at Cold Spring Harbor Laboratory in Maine, reaching the position of Assistant Director for Research under Dr. James Watson. His research activities involved the isolation and characterization of type 2 restriction endonuclease enzymes and DNA methyltransferase enzymes from various bacteria. He co-discovered DNA-based flipping in 1993, and more significantly, he independently discovered split genes and messenger RNA splicing in adenovirus system in 1977. For this work, he was awarded a Nobel Prize in 1993. One of his current interests is on the identification of restriction enzymes and methylases in DNA sequences deposited in DNA databases, and the development of rapid assays for these enzymes. So, with his extensive experience in both academia and industry, Dr. Richard Roberts is in a unique position to present his view on today's topic, spreading peace through science and commerce in developing countries. Dr. Roberts, please. Good afternoon again. It is a great pleasure to welcome Dr. Richard Roberts to this series of special lectures. Before we begin our special seminar, Assistant Professor Dr. Nippon Tuwanon, President of Chiang Mai University, would like to say a few words to welcome Dr. Richard Roberts. Dr. Nippon, please.
Dr. Richard J. Roberts, Mr. Yanis Hegwai, Director of Events Coordination of the International Peace Foundation, Professor Supot Wuttikan, Dean of the Faculty of Medicine, distinguished guests, ladies and gentlemen. 2004 marks the 40th anniversary of the founding of Chiang Mai University, and I am sure our former president would agree that one of the rare privileges bestowed upon a president is the opportunity to preside over such an auspicious occasion, as having a Nobel Laureate for medicine in our presence. Dr. Roberts, it is indeed an honor for me to welcome you to our university. Dr. Roberts is with us today under the auspices of the Nobel Prize Laureates and Celebrities Lecture Series, a program which will provide our staff and students with the unique opportunity of coming face to face with some of the world's most distinguished scientists and influential figures. A once-in-a-lifetime opportunity that I am sure will be a great source of inspiration. I would like to take this opportunity to express our gratitude to the International Peace Foundation, without whose support today's lecture would not be possible. Also to thank the Dean and his colleagues in the Faculty of Medicine for the time and energy they have contributed to organizing and hosting this event. Dr. Roberts, our most sincere thanks and appreciation to you for honoring our university with your presence. And I trust that the memories you take with you of our university and our city will hasten your return in the not too distant future. Thank you.
Our speaker today, Dr. Richard J. Roberts, is a Nobel Laureate for Physiology or Medicine and a research director at New England Biolabs, a biotechnology company based in Massachusetts. Dr. Roberts was educated in England, attending the University of Sheffield, where he obtained a PhD in organic chemistry in 1968. He did his postdoctoral work in Professor Jack Strominger Laboratory at Harvard University, studying the transfer RNAs that are involved in the biosynthesis of bacterial cell wall. From 1972 to 1992, he worked at Cold Spring Harbor Laboratory in Maine, reaching the position of Assistant Director for Research under Dr. James Watson. His research activities involved the isolation and characterization of type 2 restriction endonuclease enzymes and DNA methyltransferase enzymes from various bacteria. He co-discovered DNA-based flipping in 1993, and more significantly, he independently discovered split genes and messenger RNA splicing in adenovirus system in 1977. For this work, he was awarded a Nobel Prize in 1993. One of his current interests is on the identification of restriction enzymes and methylases in DNA sequences deposited in DNA databases, and the development of rapid assays for these enzymes. So, with his extensive experience in both academia and industry, Dr. Richard Roberts is in a unique position to present his view on today's topic, spreading peace through science and commerce in developing countries. Dr. Roberts, please.
Well, thank you very much. It's a pleasure and an honor to be here. What I'm going to do today is to talk to you a little bit about my own personal views of science and commerce and how I believe that inherently they are peaceful activities, activities that promote peace and not war. And 4 of the reasons behind this are shown on this slide. One of the principal reasons that I believe science is a peaceful activity is that scientists, no matter what country they're in, Thailand, the US, Britain, Russia, wherever, we all speak a common language. And I don't just mean English, which of course for science is probably the key language that you need to know sort of linguistically, but rather the language that we speak as scientists is one in which logic, reason, guide the way in which we reach decisions. We look around the world, we make observations, and we try to make sense of them. We try to produce theories. We try to argue in favor of our theories. We try to prove or disprove whatever hypotheses we've got. And these common goals and common language that we have as scientists means that no matter where I go in the world, I know that there will be friends. I know that I will be received very hospitably and very nicely, and I can talk to my fellow scientists very easily. One important thing here is that while we share these common goals and these common methods of reasoning, we do disagree from time to time. Not all scientists believe that a particular idea or a particular hypothesis is correct. But to the best of my knowledge, no scientist has ever used violence in order to settle a dispute. That's just not the way that we deal with disputes and the way in which we deal with disagreements. Rather, we use logic, we use reason, we try to do everything we conceivably can by force of argument to show that one view or another view is correct. I think this is a very important message that it would do well for our fellow citizens to take notice of, to see that it is possible always to resolve disputes by reason. There is rarely a need for violence in order to settle disagreements. I think as scientists, we grow up believing this to be the case. As students, you learn all about this kind of thing, that it is logic, it's reason, it's argument that will decide what is going on. So here we have common goals. We're all striving for the same thing. We all know the basic rules by which we decide things. We settle our disputes in a logical fashion by argument. And most importantly, science truly is a transnational activity. I remember back in the 1970s when I was involved in science at Cold Spring Harbor, and in the early days when we were making restriction enzymes in my labs, these were were reagents that every molecular biologist wanted. They all wanted some of these samples. They all wanted strains that would produce particular enzymes. And we used to give away these enzymes. We would give away the strains to anybody who came. We would send them to Russia. At the time, the Cold War was going on. I would go and visit Russia and would have very cordial relationships with my friends in Russia. Their scientists were often able to come and visit us, and I think it was as a result of these kinds of scientific exchanges and the corresponding cultural exchange that took place. They would find out about us in the West, we would find out about them in the East. And it's really quite difficult when you know someone face to face, when you shake their hands, to be mad at them. It's really rather easy and much easier to be friendly. And so I think a lot of what happened in bringing down the Cold War was this kind of exchange, both science, there were cultural exchanges of one sort and another, but I really believe that science played a big role here. One of the things that I was made very aware of in the 70s when I was going and interacting with Russian scientists was how easy it was to circumvent what were otherwise politically almost insuperable barriers. It was actually quite difficult to travel from the US to Russia if you were just an average citizen. The US State Department didn't want you to go, and the Russians didn't particularly want to host you. But as a scientist, it was made very easy. I would apply for a visa. I was given a visa. It was seen that I had some good reasons to go. Similarly, for the Russians, who were under even more strictures than we were, they had a very hard time getting a visa to leave Russia, mainly because the authorities didn't think they were going to come back. They thought as soon as they gave someone a visa, they would leave, and of course a lot of them did. But nevertheless, the scientists within Russia were the people who probably had an easier time than anybody else in terms of getting permission to leave. And so one of the very good aspects of science that even goes around today is that it is still fairly easy to travel between countries that perhaps other people find it more difficult to get into. In the US, a good example is Cuba. It is really very difficult to travel between US and Cuba, although it's a little easier now than it was. But there have been many scientific exchanges dating back again to the 1970s, the early 1980s, in which scientists could make this transit, could move from country to country. The third point that I have up here is the access to politicians. You know, there's a common phrase that it's really only politicians who ever go to war and get everybody going. I mean, if you look at the US and what has been going on recently this year and last year, it's the politicians who made these decisions. It's very rare that private citizens choose to go to war. And one of the things, one thing that a scientist has and that science brings, and especially if you're at a reasonably high level in science, is that you really do have access to politicians. And let me tell you one story here that perhaps gives you some idea of what can go on. In the late 1970s to mid-80s, I spent quite a lot of time traveling in between the US and Pakistan. At the time, Zia al-Haq, the dictator, was in charge of Pakistan. But as a visiting scientist with some reputation at the time, I found it very easy to actually meet with him whenever I was in Pakistan. And as a result of those meetings, he turned out to promote science quite actively in Pakistan. A friend of mine set up a major laboratory for the study of molecular biology to try to bring molecular biology into Pakistan. That lab is still running today, even though, of course, President Zia got shot down in a plane, but the science is continuing. And in fact, the science is doing rather well. And what they have chosen to do, the scientists in Pakistan at this Center for Molecular Biology, is to focus on problems that are important for Pakistan. So they're making genetically modified foods. They're focusing on diseases that are important to the population of Pakistan. And as such, they've gained a rather large amount of respect within Pakistan, but perhaps more importantly, they've been training the young people. Of course, if you watch the news or listen to the news, there are other aspects of science in Pakistan that are perhaps not so wholesome, particularly on the Atomic Energy Commission. And surprisingly, the molecular biology that is going on in Pakistan at the moment came out of that same Atomic Energy Commission. The person who runs this lab in Lahore, the Center for Advanced Molecular Biology in Lahore, he used to work for the Atomic Energy Commission. So, of course, not all is good in terms of this, but there has been some very good stuff that's come out of it. I might also add that commerce can also be very good in terms of giving one access to politicians. And another rather good example of this came recently in the problems that have been going on between India and Pakistan for a number of years now. But over Kashmir, about little over a year ago, there was a tremendous increase in the amount of rhetoric. The politicians were saying all sorts of warlike things. They were accusing one another of escalating the violence, and things were getting really very serious. What happened was that a number of companies in the US got quite worried about the situation that was going on. And the reason they were worried is because it was threatening their business. One of the interesting features of life in India at the moment is that they have focused very heavily on education. And in the south of India, in a place called Bangalore, there is a university, the Indian Institute of Technology, which probably produces some of the brightest students you could wish for. Many of those students have gone into computer science. And in the south of India at the moment, there is a tremendous amount of computer science going on. Lots of programming going on. Many US companies, British companies, French companies actually export their programming jobs to India because, of course, wages are a lot less there. But more importantly, the quality of the people who are there working in this industry is extremely high. When all of the problems between Kashmir, between India and Pakistan over Kashmir started to come up, a lot of US companies were very worried that they didn't want to get their programming done in India anymore, because if the political situation was very tense, then all of their computer programming was in jeopardy. Perhaps these people would be called up and would have to work for military purposes rather than for commercial purposes. When they were told, when the leaders of the computer science section in the southern part of India became aware of this, and the American companies basically said, we're going to take our business away, we're not going to use you anymore, they got very worried because this is an important part of the economy there. And the leaders of industry, of the computer industry anyway in India, started to talk to the politicians. And within a matter of a week or two, all of the rhetoric, all of the words that the politicians were saying, all of a sudden died way down. The situation was diffused. And so here was a case where having access to politicians from industry turned out to be very beneficial for the process of peace. Now, on this slide, I'm trying to make the point that one of the good things that comes out of science, aside from all of the common language and learning how to solve disputes, is that most of the science that takes place actually leads to applications. It leads to things that are going to be good for society. And I just put a few things here of how basic discoveries have led to applications. So within the physics community, studying the fundamental properties of optics, engineering, and so on, has led to the development of lasers, has led to the development of computers. And lots of things have come out of physics that we wouldn't know how to operate today without them. In the case of chemistry, there have been tremendous numbers of chemical products that have come, again, from basic discoveries that have been made in chemistry. I just mentioned a couple here: detergents, fertilizers. And of course, in my own field, in biology, obviously a tremendous drive behind the science that goes on in biology is concerned with human health. And so we have drugs, we have the big pharmaceutical companies that produce many drugs. We also have a tremendous impact on agriculture, crops, traditional plant breeding, learning how to take crops that were originally developed 5,000 years ago in the Middle East and making them better by breeding, pulling in genes from wild strains, making the crops better and better, have led to tremendous increase in prosperity, have led to much better health, much better lifestyles for many, many people. But of course, much of what goes on in this area goes on in the West. It goes on in the US, in Europe, and in general, I would say Asia, India, much of Africa, South America have been trailing behind in terms of what they have available in terms of the crops. But here are wonderful opportunities for anybody, any scientist who is really interested in pursuing commercial applications of really very basic kinds of research. One can work on the local crops, on the foodstuffs that are important to you, rather than the foodstuffs that are important in the West. And so I think there are wonderful opportunities here to actually exploit biology and to make some commercial applications out of it. Let me just tell you a little bit about how biology and biological research works in the US. Much of the government's money goes into this first line of research and discovery. So for instance, the National Institutes of Health spends of the order of $25 billion a year supporting research, basic research that goes towards understanding, say, the human genome, understanding organisms, understanding disease, understanding the brain, understanding mental processes of one sort and another, but all very basic kinds of activities. So in the case of, say, human biology, the US supported the sequencing of the human genome. Huge amount of money went into understanding the basic sequence of all of our genes. Now there is a great amount of money being spent to understand what these genes do. And in fact, when we look back, we see that actually determining the sequence of the human genome was pretty easy, but understanding what the genes do and how the gene products then interact with one another is much more difficult. Some of this will be paid for by private industry. So there are many drug companies who can just look at a bit of DNA sequence and know that here is a gene that may well present a very good drug target. So this kind of work you can get paid for in the private sector. You can get industry to pay for this. And so one has benefits coming out of the basic research that lead ultimately to better health. One of the other aspects of this is understanding genetic disease. You know, we all have many defects in our genes. Every single one of us in this room probably has some genes that are a little bit defective. They don't work quite as well as they should. For the most part, it doesn't cause us a problem, but for some people it does. Some people have defects in genes that lead to cystic fibrosis or lead to breast cancer or make them more susceptible to other kinds of cancers. And so this again is something where diagnosis can be important. If you can recognize these disease traits early on, maybe you want to alter your lifestyle so that you try to avoid the consequences. People who have genetic propensities for diabetes, for instance, may want to avoid various things. There is also a certain amount of money in the US that is spent on agriculture, but not very much. A little bit of research is paid for by the USDA, the Department of Agriculture, but they don't put much money into that. And the reason is that agriculture is really big business. There are a lot of very large seed companies. There's lots of money in the private sector that is supporting research in agriculture. But unfortunately, a lot of that money that goes into agriculture is spent upon crop developments rather than understanding the basics of plants. And so the USDA, the government, supports much of the basic work of trying to understand how plants work, what the genes in plants, how they differ from the genes in humans, and so on. And the last area I want to talk about here is one that is really quite interesting. It's the area of forensics. I'm sure everybody here is very familiar with the use of DNA fingerprints in order to see who's been going in various places to link them to crimes and so on. Interestingly, forensics has much better tools now than fingerprints, and these tools have all come from basic research. And the tool that most people are familiar with is the idea of a DNA fingerprint. The people, when they touch things, when blood is spilt, there are cells left behind. Someone grabs a piece of human hair, pulls out some of the cells that are there, and from these small traces one can identify who this person is. We have developed tests both in the US and in Europe, and there are even better ones coming along all the time, that can do matches of DNA samples to such a degree that any single person on this earth, with the exception of identical twins, can be distinguished one from another. So unless we have two identical twins in this audience, I don't see any sign of it, but maybe I'm wrong, but every single one of us in this room has a DNA fingerprint that is different from not only the other people in the room, but for everyone else on Earth. And this is very useful in terms of a forensic capability of identifying who was at the scene of the crime. So this is something, again, where basic research led to very interesting commercial developments. Forensics is now quite a big commercial venture in the US. And it's also something that I think is very good in terms of making the society inherently a lot better by making it rather easy to identify perpetrators and rather easy to take out those elements that cause problems in society. Now, in developing countries, obviously one has a slightly different slant on things. Many of the discoveries, many of the applications that have come from basic science in the West are sometimes rather expensive as far as the developing world is concerned, but also they're not always applicable. And I just have three different things that I want to talk, three topics I want to talk about here. One is genetic heritage. And I'm not just talking about human genetic heritage. I'm talking about all of the native animals, the plants, the bacteria, the insects, the various things that are unique to Thailand or unique to Southeast Asia. Things that really are a very valuable commodity. In many ways, they're every bit as valuable as the gold ore or the silver or platinum or other metal ores that you can find in the ground. The genetic variability and the individual genes that are present in organisms in Thailand, whether they're people or whether they're insects, can be a very valuable resource and something that I think one should, as a developing country, pay some attention to. For instance, it's probably not a good idea to be destroying the environment and destroying all of those rare species of plants which perhaps have useful products in them, useful genes that may lead to better health. You also have something going on in various countries throughout the developing world in the field of crops, in the field of agriculture. Most people here do not eat the same diet that we eat in the West. We eat some rice in the West, but nothing like the amounts that you do here. Many of the plants here, I was up in a market not too long ago looking around at some of these wonderful fruits and vegetables that you have here. Just remarkable things that we just never see in the West. And some of these are really staple items as far as the population here is concerned. Again, here, if you wanted to engineer better varieties, you wanted to engineer things that were easier to grow, or even to engineer things to put in genes that would make these things a lot better. That's something that you can do here that's not going to be done in the West. These are not the kind of problems that typical Western scientists get into. And then finally at the bottom here, you have many different health problems. Obviously, we all have problems with common diseases like the common cold and flu and so on. But there are many other kinds of health problems that really affect the developing world that do not affect the West in any or at least not yet. Some of the tropical diseases, malaria is not a problem in the USA and Europe and so on. Fortunately, it's not too much of a problem here in Thailand. But for a lot of the developing world, malaria is a major problem. 1 million people a year die from malaria. This is a problem that in the West we have tended not to tackle. The only reason typically that the US ever gets involved, Europe for that matter, ever gets involved in tropical diseases is because they want to send soldiers over here to fight and they want them not to get sick with all of the diseases that are endemic. That's really not a good reason to be doing anything. I think this is something that is a great challenge and great possibility for the developing world to get involved here. And I'll tell you later about one particular disease that my company has been working on that illustrates, I think, what can be done if you really get involved in this area. There are also genetic diseases that typically affect different parts of the developing world, things that we just do not see in the US. So for instance, in China, there is quite a problem with a virus, Epstein-Barr virus, which actually causes a cancer if you have the incorrect genes, as it were, in your body. And so this Epstein-Barr virus, which when it infects someone in the US, typically just causes a disease which we call mononucleosis. It's sort of a tiring disease. You maybe get very tired and have to sleep a lot. But it comes and maybe you have it for a month, maybe you have it for a couple of months, but then it goes away and you're fine. That same virus in certain segments of the Chinese population causes a particularly nasty cancer, very nasty. And in other populations, it causes Burkitt's lymphoma, a different kind of cancer. The first one was nasopharyngeal carcinoma. So there are different health problems that are faced in the developing world than those in the West. And again, in general, these problems, the West don't care about them. They're not going to work on them. But these are open opportunities here for scientists in developing countries. Now, what I want to talk about now is how we in the West can actually help scientists in developing countries and how through science and through commerce we can hope to spread this message of peace that I'm talking about. The first thing we can do is to make sure that we give lots of opportunities for scientists to be trained in the West. This has been a program that has been going on in the US and in Europe for many years now. There have been certain problems with it. A lot of the people come over, get trained in the West, and then never go home again. They find a good job in the West, and so we train them, but the benefit back to the developing country doesn't come. However, I think things are improving now because as the developing countries themselves are prepared to put in some resources to try to keep the scientists in the country after they've been trained by presenting job opportunities and so on, and this can be really useful. So I think there are some very good possibilities here, and it's actually perhaps easier than one might think to get a good training in the West. Having trained them, of course, and you send them home, they need to continue to get support. And while resources in the developing world may not be as great as they are in the US, very often just small infusions of materials, of research materials, of ideas, and so on from a Western lab can make a big difference. And my own experience has been that by establishing individual contacts between scientists in a developed country and a developing world, you get to know people, it becomes easy to exchange materials, and you can do things that ultimately will benefit both labs, and I will show you an example of that in a moment. More importantly, once commerce gets underway as a result of the scientists that goes on in a developing country, one can offer market opportunities. And again, I will show you on a small scale how that is possible with the example of my own company. And finally, one of the things that I think we have a responsibility to do in the West is to encourage the further development of scientific research within the developing world. And so to this extent, it's always nice, I find it very nice to come and talk to people of some importance, university leaders and so on, to encourage them to really make sure that the scientific opportunities are present for students like yourselves, so that once you get a good idea of what science is all about, that the opportunities are there to expand it, to move into commerce or to move into further education. Now, what I'm going to do now is tell you a little bit about the company for which I work, New England Biolabs. And we're a very unusual company. We're not like any company, I suspect, that you've ever heard of. The company began in 1975 when the owner of the company, a man called Don Combe, was a professor at Harvard, actually an associate professor at Harvard Medical School. Harvard Medical School, very, very prestigious university medical school. And they had a policy in certain of the departments that they simply would hire young people into the departments, but then they wouldn't give them tenure. They would be there for 5, 7, 8 years, but then they would have to leave. Now, Harvard argued that this was a good thing because they got lots of turnover, lots of good young people coming in. Typically, people took these jobs because they thought they were so good that by the time the 5 or 8 years was up, they would have made some major discovery. The department would have to give them tenure because they were so good, and so they were going to be the first in the department ever to get tenure. So there was their ego at stake. But then when the tenure decision came and they were told they had to go, they rationalized it and they said, well, you know, no one ever gets tenure in this department, so I'm no worse than anyone else, and so they went off and got a good job somewhere else. Well, Don Combe, the man who started Biolabs, said he didn't like this system. He didn't think this was the way to do science, and rather what he was going to do was to start his own company, use the profits that could be made from that company to support research, and build up a small research enterprise that was basically supported by the profits from whatever the company was doing. And in this way, he thought he could build a first-class research enterprise that didn't rely upon government funds or anything else for for their funding. It just came from the profits from the company. So we thought we could make Harvard and NIH all in this one institution. So he started off in 1974, actually in late 1974, just making a few reagents for research, things that research scientists need in order to do their work. It's sort of like if you're in a chemical lab, there's a few chemicals that you have to have. In a biochemical lab or molecular biology lab, there are fewer reagents that everyone has to have. And so he was struggling along doing this for about a year, but not making enough money to support the company. The company at the time was Don, his wife, and one technician. So there were basically 3 people. So he did a little lobster fishing on the side and some pregnancy testing just in order to make ends meet. And then he had the idea that perhaps it would be good to sell restriction enzymes. And so he contacted a company called Miles, who basically had a big catalog and they sold all sorts of stuff to researchers, and asked them if he made some restriction enzymes, would they sell them? And they said, well, you know, we'll put it in the catalog and just send us some. Well, it was about that time that I, at Cold Spring Harbor, had been trying to get Cold Spring Harbor Laboratory to set up a company specifically to make restriction enzymes, because we discovered some 20 or 30 of them in my laboratory at that time, and everybody wanted them. We were giving them away. We were sending them all over the world. Everybody wanted them. And I thought, what a great way to get some money into Cold Spring Harbor Laboratory, which is a private research laboratory. But, it's a not-for-profit thing. Cold Spring Harbor relied upon government grants and so on for all the work. But I thought, why don't we just set up a company and have all the profits from this feed back and support the research at Cold Spring Harbor? And so I spoke to my boss at the time, who was Jim Watson. This is Watson who discovered the DNA double helix. And I said, I got this great idea. Why don't we do this? And he said, oh, you can't make money doing that. Plus, at that time, it was considered not quite the right thing for scientists to be doing, sort of to get involved in commercial ventures. And so he wanted no part of it. He didn't think it was a viable proposition, and he thought it somehow was slightly a dirty activity. So I looked around, and eventually I was told that this company, Miles Laboratories, were thinking of selling restriction enzymes. So I called them up. Let me just take a drink of water here. This is where we need some of the expertise from the medical faculty to take care of this infection I have. So I called Miles Laboratory, and they said, well, it's not us, it's this guy Don Combe. You should go and talk to him. So I called him up. He said, come on up, let's talk about it. And as we say, the rest is history. The company started to sell a lot of restriction enzymes. And at this point, the company is about 210 people. We make about $60 million a year. We support a research activity so that half of the company is doing research, the other half of the company is making money. The scientists who work at the company have a pretty free hand to do what they want. All we ask of them is that 1/3 of their time is spent doing something that the company thinks is interesting. That can vary from working on something that's going to be a commercial product to working in some area that we have deemed to be important. One of the things we decided right at the start was that in addition to just doing basic research that might be of general interest, we would also try to find some research topic that would be useful within the developing world. And we, in 78, 79, had a look at the top diseases of the world that the World Health Organization was concerned about, picked one that didn't have a lot of funding and which we thought perhaps recombinant DNA could be useful for, and started working on it. That disease was filariasis. Filariasis, you probably have heard of elephantiasis or onchocerciasis, river blindness. These diseases are caused by small parasitic worms, live in the lymph systems, and produce microfilaria, which are sort of the babies of these worms, and they cause all sorts of problems. It's a very nasty disease. It's endemic throughout the tropics. The total world funding for filariasis was probably about $100,000 a year in 1981, 82, when we first got started seriously doing this. We've been spending initially about $1 million a year. Now we're spending $2 million a year studying some basic aspects of this disease. The idea was just to do some molecular biology, see if we could find, using recombinant DNA molecular biological techniques, things that would be useful. Not necessarily just for us, but things that would be useful for anybody who was interested in working on this disease. We started by looking for surface antigens, proteins that were sitting on the outside of the worms that might be useful in terms of developing a vaccine. The intention was never to make any money at this. This was done purely for humanitarian purposes because we thought it would be a fun intellectual project to get involved in. That work has taken a number of interesting turns, the most recent of which is that a little over a year ago it became apparent that these worms had living within them a small bacterium that was living inside the cells. And if you kill this bacterium, it's called Wolbachia, if you kill the Wolbachia, you kill the worm dies. Now bacteria is something that the drug companies really know how to deal with. They have many antibiotics. The problem is to get something that will actually get through the worm, get into the Wolbachia and kill it. And so our most recent venture has been to actually determine the complete DNA sequence of the genome, the DNA genome of this Wolbachia. And as a result of that, we actually have several incredibly promising drug targets, that is, genes that are clearly of crucial importance for this Wolbachia to live, but which are not present in humans, and not present in the organism, Brucia malayi, which causes the little worm that causes filariasis. And so now we're trying to work with some entrepreneurial drug companies to see if we can find a potential drug that might be useful. Of course, we're never going to make any money out of this because it's a disease of the developing world. It's not a disease in the US. But I say that really wasn't our aim in the first place. But quite by chance, as a result of this work, we have made a lot of money. And it turns out that some of the antigens that we found on the outside of Brucia malayi are also present on the outside of Dinofilaria, which is the worm that causes heart disease in dogs. It's a dog heartworm. And of course in the US and Europe, loads of pets. Everybody loves to have their pets. And they're not very happy when these dogs have heartworms or when the cats have heartworms. And so they will take them to the doctor and we'll get a treatment for them. There are various treatments for these heartworms. But you don't want to give the treatment unless you know the dog has heartworm. And so it turns out that this one antigen is a great diagnostic for dog heartworm and for cat heartworm. And a company that was making these diagnostic tests came to us and said, you know, we'd be interested in exploiting this. And so we ended up making quite a lot of money, not for filariasis, but for treating people's pets in the US. So quite by chance, it turned into a commercial possibility. You'll see down here I have a list of countries in terms of things that we're doing in the developing world. One of the things that we started to do when I was first associated with biolabs was to screen bacteria to find new restriction enzymes. Restriction enzymes, you'll remember, these were the things that got the company going. Well, screening bacteria is pretty tedious work. It's very straightforward, very easy to find these things, but after a while it gets a bit tedious. And so we decided that what we would do is we would take this opportunity to get some scientists to come to the company from developing countries. We would teach them how to do the screening, send them home, and let them set up a small lab where they could screen and try and find some new restriction enzymes for us. Any they find, we get the strains, we sell them, and we give them a royalty on anything that comes back. And we also support their labs. So we now have labs in Cameroon, in China, in Nicaragua, in Portugal, in Uganda, and Vietnam, all of which are busy screening for restriction enzymes for us, and for which we provide support. We bring the scientists over to New England Biolabs, we train them, send them back, help them get their labs set up, and then we send our scientists over to visit them and help them out in their home countries from time to time. Anything they find that is commercially viable, we will sell it and then send a royalty back to the lab. So I think this has been really a very nice way for us to get involved in a whole bunch of different countries. And I think this kind of model, there are many companies that could do exactly the same kind of thing. This isn't something that is unique to what we do. There are many opportunities for this kind of thing. And so if any of you ultimately think, well, maybe you'd like to start your own little company based on science, think if there's not an area you can get into where you could find a company in the West that would help on something like this. Oh, this is the filariasis. I've talked about that already. So, what I've tried to do in this talk is to give you some idea of how science and commerce go hand in hand. Science is this universal language, this universal discipline that I would argue is inherently peaceful. But I think commerce, too, is also an inherently peaceful activity. Businessmen don't like to have wars going on. They want a nice, stable environment in which to conduct their business and to make money. And furthermore, if you're in a position where you're involved in commerce in some way and you're making some money, there's a much greater chance that you will be happy within your society. It very often tends to be the people who have nothing or the people who are being treated badly within society that decide they're going to become militants and cause problems and so on. But if there is good commerce going on and science offers at least one way in which you can have good commercial prospects, I think the chances of having a peaceful society and ultimately of having peace between societies when they sell their products to one another are much greater. And so my premise is that both science and commerce are inherently very peaceful activities that tend to promote peace and offer ways in which you can solve problems by talking about them, by discussing things, by using logic and reason. And I guess the point that comes out of all of this is that when we look at the people who are making decisions in societies at the moment, they tend to be politicians. Most of these politicians have not come from a scientific background. They're unfamiliar with the methods of science. They're unfamiliar with the logic and the reason that leads into science. And perhaps what we should do is to make sure that we get many more scientists involved in decision making and get many more scientists into these leadership positions. At my company at New England Biolabs, everybody who makes a decision at that company is a scientist. We have no businessmen in the company. Only scientists make decisions. Almost every time when I talk to business people about the way in which we've chosen to structure the company and to run it, they tell us we're crazy. If they were a businessman, that's not the way they would run it, because all they're interested in is making money. And we're not interested in that. We're interested in making enough money to support our science. We're not interested in just making the money per se. And so I think there's a lot to be said for getting the logic and the reason that goes behind science, the fun that can go into science, involved in a much larger way in helping to spread peace around this world. So I thank you all for your attention, and I'll be very happy to take any questions you might have.
That's a very interesting views on science, commerce, and peace. And we have a few minutes for comments and questions for Dr. Roberts. And please feel free to use either English or Thai, and I'll be happy to translate for you.
And I think you should feel free to ask any questions you want. It doesn't have to be about anything I talked about.
Dr. Roberts, I'm very sorry every time I heard that the Western country doesn't produce any drug that's useful for treatment of infectious diseases. That's very important for the East. But it's nothing means nothing to the West. But today, I think, after his talk, I feel very happy that at least some company pay attention to the drug that for treatment of filariasis. That's very unique to the East, but nothing means nothing to the West.
Unknown in the West, right.
So I think the same as Deposy. I'm working in Deposy for more than 10 years. And the West company, they never interested in the drug for treatment of Deposy. So I think from your talk, is it possible to create an international institute to do anything that benefit to the East and to the West, both. Not just concern the West.
Yes, leprosy is actually a rather interesting example because there has been a tremendous amount of work done in the West to sequence the genomes of bacteria. Bacteria that cause disease have been a particularly high target. And so as you're probably aware, the genome of Mycobacterium leprae, which causes leprosy, has been sequenced at the Sanger Center in England. And it's a very interesting genome. The genes look as though they're in a real mess by comparison with other mycobacterial genes. There are some very interesting scientific findings there. But I believe there are also some potentially interesting and good drug targets that are present in leprae. I don't know what the follow-up on that is, but so I think the basic tools, the basic genomic tools, and there is a lot of knowledge now about leprae that they didn't used to be before, and one can only hope that that will have some useful impact later on.
I raised the question, I think, can you do this I heard a lot about the International Research Institute. They will do any research for the world, but that's just only the name. I want to see the real international institute that they can do research for both the East and the West. I think you may be in the position can do this job. So, so okay, thank you.
Okay, well, I'll do what I can.
I'm from a pediatric dentistry department, Chiang Mai University. Just want to ask you, the topic today is about peace. I'm wondering, you got the Nobel Prize in medical science, right?
Yes.
And what made you choose the topic to talk about peace instead of what you
so
got the Nobel Prize
I was
for?
invited to come here by the International Peace Foundation. It seemed appropriate to at least offer to talk about peace a little bit.
Okay, that's answered. And could you tell us, or students here, the young students here, about what did you do to get the Nobel Prize, and what should they do to not to be that doesn't have to be that good, but just to encourage them to be good in science. Thank you.
Okay, so I won the Nobel Prize for discovering that genes in humans and all higher organisms are different in the way in which they're organized from genes in bacteria. Genes in bacteria have a linear piece of DNA. You start at the end, you read it, you get to the end, and that is the gene. It codes a protein. You don't need to do anything special. In humans, that's not how genes are. Genes in humans are split up into pieces. They're typically spread over enormous segments of DNA. There's a little bit here that it makes some sense, a little bit here that makes sense, a bit here that makes sense, and so on. And all of these pieces have to be rejoined before they can be translated and made into proteins and so on. Prior to 1977, everybody thought that all genes in all organisms look like bacterial genes. What we discovered in 1977 was that human genes and all higher organism genes are really very different, and they're split up into pieces. And the way in which we made this discovery was that I was interested in what the start of a gene looked like. We knew in bacteria exactly what the start of a gene looked like, and everybody had assumed the same would apply in higher organisms. The starts of the genes would be the same. And it was as a result of trying to understand this that we discovered the gene was all split up. So it was a serendipitous discovery. We were trying to do one thing. We got data that didn't make any sense, came up with a theory about what must be going on, spent a year trying to prove what we thought was going on. Eventually we did the experiment and ended up discovering split genes. And my advice, I think, to all students is don't aim to win a Nobel Prize. Don't aim to do something absolutely extraordinary. These things tend to come as a matter of luck. They come because you have a well-prepared mind. You look carefully at what's going on. But the most significant thing you can do if you want to have a happy and productive life, don't try and find a job that's going to make a lot of money. Find a job about which you can be passionate, that you like it so much that when you go to work, it's not like going to work, it's like pursuing your hobby. So find whatever it is you're really passionate about and make a career doing it. I don't think I work. It's never occurred to me that what I do is work. What I do is I pursue my hobby. I love science. At the moment, I'm really into computers, into computational biology, love sitting in front of a computer doing stuff. I don't view it as work. I love Monday mornings. I can't wait for Monday to come around so that I get back and get into the lab and begin to do things. And if you do that, you will have a very happy life. Money doesn't bring happiness unless you really enjoy what you're doing in life. So find what it is you really love to do, you can be passionate about, make a job of it, and you'll have a really good life. But don't worry about the money. Money, if you've got a good job, you're okay.
Actually, Dr. Robert, please.
I hope no one objects to a Falang speaking. Firstly, may I congratulate you, sir, on your talk. I found it extremely interesting, but I want to add, if I may, a caveat. I think you were talking about good science.
Mm-hmm.
And I'm one who does believe there is bad science. One of the things you emphasized was training. And indeed, in terms of genetic heritage, Thailand has suffered greatly through the training its agriculturalists received in Europe, I can say that as an Englishman, and in the USA, and I can say that because you represent both. Because, for example, Thailand has operated an agricultural policy in livestock which worships the exotic, mostly because its scientists in genetics were trained in Europe and the States. They were trained in, if you like, short-term commerce rather than good science, which was the background to why certain genetics are good in certain situations. I have one question, and it centres on the fact that Thailand is very similar to the USA in one context. Both have failed to ratify the Convention on Biological Diversity. My question is, how do we scientists get our politicians to think sensibly on such matters?
Well, I certainly agree with your caveat that it's good science that one needs to promote. I think the question of how you get politicians to behave properly is much more complicated. I think one of the first things to do is to elect them out of office and get rid of them, which would certainly be my solution for President Bush at the moment. And as I tried to say during my talk, I think scientists often run things a lot better than politicians do. And so maybe we should try and get more scientists into politics. Now, there is a caveat to that, and that is that a lot of the scientists who get interested in politics don't end up making very good politicians. There are many medical doctors, for instance, who decided politics is a good thing to do, but when it then comes to making decisions, they don't really seem to take their medical training into account. Maybe the ones who go into politics are all bad doctors to start with, but certainly the Senate Majority Leader, for instance, in the US Congress at the moment is a doctor, but I've not seen any evidence that he's been making wise decisions as a result of his medical training. Perhaps the real answer, though, is that we should only have politicians who don't want to be in politics. I think there's a lot to be said for electing people to leaders who really don't want the job. If people want the job, they usually want it for all the wrong reasons, and as soon as they get it, the only thing they're interested in doing is trying to make sure they get re-elected rather than really trying to make sensible policy. But these are just a few vague ramblings. I don't have a good answer. I think, we have to try to find better ways of getting better politicians, higher quality politicians leading our countries.
I was about to say that actually Dr. Roberts has presented his work, Nobel-winning work at Chulalongkorn University, and his new work on base flipping at Khon Kaen University yesterday. And if you are interested in more in his life as a scientist and his work, we can follow up on his autobiography that is available at the website of Nobel Foundation. Right. So I think because of the time limitation, I would like to close the session, and I would like to invite Professor Dr. Supot Buttikan, Dean of the Faculty of Medicine, to again express our gratitude to Dr. Roberts.
I would like to say a few remarks to Professor Richard Robert. In behalf of the Faculty of Medicine, Chiang Mai University, I would like to say many thanks for your contribution this afternoon. Thanks for the International Peace Foundation who support this event. Since the President of the University has said that this year we celebrate the 40 years anniversary of the university, that's why we invite the Nobel Prize Laureate to give a lecture. I expect that you will come back when we celebrate the Golden Jubilee anniversary. And lastly, I think it's not too early to say Happy Valentine to you. Thank you very much.
Now I would like to ask the President and the Dean to present tokens of our appreciation to Dr. Roberts on the stage. Now, I would like to invite our faculty members for a photo session on the stage, please.